Welcome to our YouTube channel, wakissha Gerald-Biologyconnect. Today we are going to share on concerning wakissha joint mock examination, Uganda Certificate of Education Biology Paper 2. And I request, as we have always been, we pay attention to this, we watch, subscribe, like and share. This paper has 2 items, that is item 1 and item 2, and here we are starting right away with item 1, which says that James and Simon are farmers in Kiichi village who planted Irish potatoes at the same time during the last planting season, and they both applied the same type of fertilizer. James used the recommended concentration and his plants grew healthy and strong, producing a good harvest. However, Simon used twice the recommended concentration, thinking that it would boost the yields. Unfortunately, at a flowering stage, most of his plants withered and the harvest was poor. We provided with the 2 solutions A and B, which are of different concentrations of the fertilizer by the 2 farmers, and specimen Q, which is a plant part obtained from the plants grown by the 2 farmers. Design and carry out an investigation, a scientific investigation, solution A and B, using specimen Q, and use your results to explain why Simon had a poor harvest and advise the farmers on the correct concentration of the fertilizer to be used. We are producing the aim of the hypothesis and variables. Note that we don't have the apparatus, meaning they are to be shown in the procedure and results, and then we have the analysis, explanation and recommendation. So here we have our guide, and we see that solution A was a 0.1 molar concentration. Something small. It was a 0.1 molar solution, that is solution A, of sucrose according to the materials that were given, and then B was distilled water, specimen Q, and Irish potato. So we are to design the experiment. So what is the aim of the experiment? The aim of our experiment, if solution to investigate the effect of different concentrations on a solution. To investigate the effect of different concentrations of a solution, that is a fertilizer, on a plant tissue, which is an Irish potato in this case. Hypothesis: if a specimen Q is placed in a solution of a higher concentration than its cell sap, it will lose water and shrink, but if placed in a solution of lower concentration than its cell sap, it will gain water and become tagged. You recall the concept of solutions of different concentrations, where I looked at hypertonic, hypotonic, isotonic solutions. So a solution, if you get a plant cell and you place it in a solution, if that solution is less concentrated than the cells of the plant cell, that one we call it a hypotonic solution. And if that solution is more concentration than the cells of the Irish potato placed, it means it is a hypertonic solution. But if the solution and the Irish potato have the same concentration, that one becomes an isotonic solution. So what are the variables? We have independent variable, the one on which the whole experiment depends, that if we are keeping on changing it to, is the one majorly, the factor that is majorly tested here, difference in concentration, is the independent variable. And then the outcome or the result of the experiment is the factor called the dependent variable, and that is the change in mass. You can study that experiment using change in weight or change in texture of specimen Q. Then you control the variables, the size and mass of the potato cylinders, things you have to keep constant. The mass, if you measured the mass first, you have to measure the same mass for equal masses for all the potato cylinder, the two you are using. And then if you're measuring maybe the length, you may have to make sure that the size is equal. Equal sizes for both of them. Yes. And then the volume of the solutions should also be equal. The solutions you're using, the duration of the experiment, and the temperature, all those should be put to constant. So the procedure is over here, using a cork borer. Cut two identical cylinders from specimen Q. Like I will show you a cylinder over here, the one I drew here. Then trim both cylinders to an equal length. In this case, for me, I used 5 centimeters using a knife. You can see names of the apparatus used, they are coming out. So why specifically we didn't even have them in the on the list they are up, so we shall be tracing them from here. So label 2 test tubes A and B and pour 20 centimeters tubed of solution A into test tube A and 20 cubic centimeters of solution B into test tube B. Then press the potato cylinders in each test tube and leave the setup for 30 minutes. Now this is a controlled variable that 30 minutes we put for A and B. They should stand for 30 minutes before we use them. We observation, then use a stop clock to time the duration. You see here another apparatus. Then after time elapsed, remove the cylinders and blot them gently with the paper towel. Another apparatus, and then measure the final length. Maybe there you can say using a meter, using a ruler. So those are the results. So that, for example, if my initial length was 5 centimeters, then the final length, initial length is 5 centimeters. 5 centi. These values are in centimeters. Same applies here. So we have to be centimeters, change in length, centimeters still. So if the final length after the reaction, for me I measured using length, another one might have used weight, then the one in solution A, which we said is a highly concentrated solution, 1 molar solution, meaning that one has more sugars, more concentrated, and it will absorb water from the Irish potato tube or Irish potato cylinder, making it to lose water and shrink, reducing in size. That's why you see the change in length is negative 2. It has reduced in size. When you say final length minus initial length, you remain with this negative here. That's why we are saying cylinder in solution A is shorter and flaccid. Cylinder in solution B is long and tugged. Why? For it absorbs water and becomes long and even hard. The values I've recorded here are not practical, but though they follow the trend we are trying to explain. So analysis: the cylinder in solution A lost water and became shorter and flaccid, while cylinder in solution B gained water and became longer and tugged. Why? Let's continue. Why? Because this is due to osmosis, the movement of water molecules from a region of higher water potential to a region of lower water potential across a partially permeable membrane. In test tube A, the solution had a lower water potential than the cells of specimen Q. Consequently, water moved out of the cells of specimen Q and into the surrounding solution, causing the cells to become flaccid and the cylinder to shrink and reduce in size. This mirrors what happened to Simon's plants, where the high concentration of the fertilizer caused the plant cells to lose water, leading to wilting and poor growth. This is exactly what we have been talking about. That because the concentration of the fertilizer you have poured on around these plants is high, there is less water in that fertilizer, then more water will be absorbed from the tissues of the plant themselves, and they shrink. In test tube B, the solution had a higher water potential than cells of specimen Q, meaning solution B is less concentrated. Moreover, it is just distilled water, meaning it has more water than the Irish potato. So water moved from a solution into cells of specimen Q, causing them to become tugged, and the cylinder to expand and increase in size. So this is similar to James's plants, which were in a well-balanced environment, allowing cells to maintain turgor with enough water that supports them upright and used in other processes which kept the plant healthy. You can see all these healthy are leading to a good harvest. So like things of that sort. So an advice to that farmer, to those farmers, I advise farmers to always use the recommended concentration of fertilizers. For instance, in this case, you can use fertilizer B, is the best. Using a higher concentration will not boost yields, but you will instead use the plants to lose water, cause plants to lose water through osmosis, leading to wilting of the harvest. Let's go back. What was our hypothesis? Our major concept in the task, and the carry out investigations using solution A and B and specimen Q to explain Simon's poor harvest and advise farmers. So we have seen what caused Simon's poor harvest and an advice to farmers whether sorry, the actual question was here, maybe we can check that to see whether we have exactly met what they meant. And use your result to explain why some monad are poor harvest and advise farmers on the correct concentration of the fertilizer to be used. So the correct concentration of the fertilizer to be used was supposed to be that fertilizer A, B, like we have seen. So I advise farmers to use the recommended concentration of fertilizers, and in this case we advise them to use fertilizer B, which because it is the one with the recommended concentration. Solution. Saying solution B is the recommended concentration. I'm trying to write this. Recommended concentration. Abbreviating this is the recommended concentration of the fertilizer that they should use. So on item 2 was on plant tissues, specifically the leaves, and Mr. Okello, the farmer, had his garden, and in his garden in the middle of the garden he had plants with leaves which are broad and the network veined. We are seeing there specimen AR, which is a bean leaf, and specimen AT, which is a sweet potato leaf, and then specimen and around in the besides around the garden leaves at the edge of the garden he planted there plants with narrow leaves, narrow veined leaves at the edge. So you're provided with the specimen R, bean leaf, specimen S, maize leaf, and specimen T, sweet potato leaf. And okay, and you're required to identify those plant organs. So number one, when we are identifying those plant organs, we have to use, excuse me a little, yes, so when we have to use their name, these groupings, compound leaf or simple leaves, or different classes of simple and compound leaves. So here, for example, bean leaf is a compound trifoliate leaf. Why compound? Because its leaflets are divided into its lamina is divided into different leaflets. Why trifoliate? Tri means 3, foliate leaves, meaning it has 3 leaflets per leaf. Amazing. Leaf, which is specimen S, is a simple leaf. Reason, it is not divided into many leaflets. Then specimen T, still sweet potato leaf. Here you have to be careful. Most of these leaves of the sweet potato, if you're not so vigilant, you may call it a compound leaf. Why? Those who do not differentiate them, you find a leaf like this, and you say because you're seeing those things, it has become a compound leaf. These ones, this is just a margin that is either wavy or crenate or what, or dented, but it is not what? It is a simple leaf. Then here, what B, they asked us adaptations of specimens from the middle of the garden. The specimens from the middle of the garden was R and T, and we say that is a bean leaf and the sweet potato leaf. How are they adapted? One, they have a blood flat shape. That is, blood flat shape is a feature, tick. And why that? It provides a large surface area for maximum absorption of sunlight for photosynthesis, another tick. In the same manner, they have a thin structure that allows for short diffusion distance of gases, carbon dioxide and oxygen, to enter and exit leaf cells, and for light to penetrate to all photosynthetic cells. They have an extensive network of veins which provides a strong support system for the broad leaf, and as well efficiently transporting water and nutrients to the leaf cells through the xylem and sugars away from the cells through the phloem. So yeah, the network vein, like I've shown here, these network veins in that leaf here, they help, those are extensions of the xylem and phloem tissues from the branches and then to the leaves, and those ones, the phloem tissues, help transport manufactured food, and the xylem tissues transport water. And finally, part C, drawing and labeling. Our drawings should have a title, that is what we are saying, that a leaf a drawing of leaf of specimen, drawing of specimen S, which was the leaf of maize. The drawing should have an outline, and when we talk about an outline, we don't mean you to draw a scale, when we say draw an outline, what other people do, instead of drawing an outline, they draw for us this one we are putting on maps frame. Here we don't talk about a frame, but we mean if it is this leaf, it should be having that outer line that is tracing. Now there is something. If you leave it that way, it will not be a complete outline. So what we should mean by complete, an outline should be complete at every point, showing exactly what this leaf is. That its outer line complete like that. That's what we call an outline. So if this was my drawing, then it would also be questionable. Our lines should be straight and one line, and it should be freehand, one line, sharp pencil, very neat, so that you don't make your diagram untidy. So that's what I'm trying to mean. Remove this one so that you can see the picture very well. You can see our picture very well. Okay, another thing is that should be having labels, should be labeled, and labeling lines should be not crossing each other, and to be neat and clean. You can see how I put them in one direction and very organized. It can go to another direction, but what is so important is the organization. We have here leaf apex, which is the upper point, that narrow point of the leaf. We have the parallel vein. This parallel vein is running down from this ligule to the apex. We have the midrib, that main vein that moves from down here to the leaf sheath and moves upwards. And then we have midrib leaf margin, which is this outer line here, the outer margin. The midrib, I said the midrib is that very inner vein, the innermost big vein that runs from down to the top. Then the leaf blade is this whole narrow passage, this whole leaf lamina is what we call the leaf blade. And then the leaf sheath is that stalk. For monocot leaves, we don't have solid leaf stalks, we just have what they call a leaf sheath. And here they put a note for us that the drawing should have the following title, outline, magnification. It should be accurate and neat. If it is a leaf, it should be a leaf, not a hide. Yes. Magnification, here someone will say you're magnifying, but it is reducing. When you look at the size of this leaf, it doesn't need any magnification. In normal circumstances, the maize leaf is big enough. Actually, to fit on a paper, it is even reduced in size instead of being increased. So that's why you see that my magnification is a fraction. Size of the image over size of the actual object. or that one causes it to be a fraction. Okay, thank you for watching. Keep watching and share a link to your fellows so that they may also partake of what you share and understand what you always share here, so that next time they may be part of us. We wish you the best. For now, we'll meet in our next video. Thank you.